One of the first, most important, and simplest parts of the work lab is running PCRs, or a polymerase chain reaction. In this video, I'd like to explain one use of a PCR and how this technique works. A PCR, or polymerase chain reaction, is a reaction that is able to make many copies of a small segment of DNA so that if that piece of DNA or gene is present, it will be amplified until you can see the product with the naked eye. The first step to doing this is by denaturing the DNA. This means that you unwind the two strands of DNA, breaking the bonds that hold these two separate strands together. This is typically done by heating up the DNA to about 94 degrees Celsius. The next step is called annealing, where primers are attached to the beginning and ends of the gene the researchers want to amplify. Primers are small segments of 18 to 30 base pairs that specifically attach to the beginnings and ends of whatever gene is being amplified. Without primers, the polymerase, a natural enzyme that adds base pairs to a growing DNA strand, wouldn't be able to make a copy of the gene. Think of the polymerase as the part of the zipper that you pull up and down to zip a jacket up, aka the pull tab. The two smooth pieces at the bottom of the zipper, aka insertion pins, act like the primers that allow the polymerase, or the pull tab, to attach to the DNA, also known as the zipper teeth, and add nucleotides. It is important to understand that one primer binds to each strand of DNA. The third and final step of PCR is called extension. In this phase, Taq polymerase, a special enzyme that is able to withstand the high temperatures of annealing, copies the DNA strand by adding more base pairs to the primer. These three steps are then repeated over and over until at least 1 billion copies have been produced, and the target DNA is visible when separated from the other DNA. In the work lab and many others, it is necessary to determine what genes are present in specific mouse subjects so that researchers can confirm that their experiments are being correctly run. For instance, mice are tested for the TWIST1 gene, which is the specific gene the researchers are studying. They also test the mice to confirm that they have the TDT, or tomato gene, which allows researchers to visually see which cells in the artery come from smooth muscle cells. These smooth muscle cells are the cell type that TWIST1 is being studied in. You can see in the image below an example of a PCR that is run on TDT, or tomato gene. It's not important to understand what these genes are for the purpose of the PCR, but I am using them as an example for why people use PCR. If a PCR is incorrectly run, and it leads researchers to inaccurately believe that one of the mice they are testing has different genes than it actually has, this can cause confusing and inaccurate results based on the data they collect down the line.